Good afternoon, distinguished guests, ladies and gentlemen, and our SUTD family. Thank you for joining us today. Before we start, a little safety announcement. The emergency exits are located at the sides of the auditorium, the center, and the back. In case of emergency, please make your way calmly, as calm as you can, to those exits. Thank you. Well, a very warm welcome to the Singapore University of Technology and Design. My name is Dr. Dawn Koh. I'm a senior lecturer from the Science and Math cluster with joint affiliation with the Engineering Product Development pillar. And today, I'll be your MC. A little bit of a background. SUTD is Singapore's 4th public university, and we'll be celebrating our 10th anniversary next year. At SUTD, we incorporate the art and science of technology and design into a multidisciplinary curriculum and seek to nurture technically grounded leaders and innovators to serve societal needs. Well, in the area of healthcare, SUTD actively collaborates with strategic partners in industry, government, and academia for education, research, and to spur innovation and enterprise efforts in the area of medical technology. One of such collaboration is the SUTD Duke NUS Special Track with the Duke NUS Medical School. Well, this program aims to nurture future clinicians, innovators who are proficient at both practicing medicine and harnessing technological advancement across disciplines to impact healthcare. Well, you're here today for the SUTD Iconic Voices from MIT lecture series that features leading faculties from MIT and provides a platform for interaction with the distinguished experts in the area of science, architecture, technology, and design. Past speakers include the President Emeritus of MIT, Dr. Susan Hockfield, Nobel Laureate for Physics, Dr. Jerome Friedman, and most recently, the Addison of Medicine, Dr. Robert Langer. Well, this Iconic Voices from MIT lecture series is proudly sponsored by Far East Organization. Our speaker for today, Professor Susumu Tonegawa. He is the sole winner of the Nobel Prize for Physiology or Medicine in 1987 for the discovery of genetic principle for the generation of antibody diversity. He has since been making seminal contributions in neuroscience. Using advanced techniques of gene manipulation, Professor Tonegawa is now unraveling the molecular, the cellular, and neural circuit mechanisms that underlie learning and memory. His studies has broad implications for psychiatric and neurologic diseases. His recent studies conducted was by combining both the molecular genetic and optogenetic technology have revolutionized memory research and was recognized in 2014 by Smithsonian American Ingenuity Award and as one of the top 10 discoveries by the journal Science. Well, the topic for today is how do we form, store, and retrieve memory. The format will comprise of a 45 minutes lecture followed by a 15 minutes Q&A session. So it gives me now great pleasure to welcome Professor Susumu Tonegawa. Professor, please.
So, good afternoon. I'm truly excited about having this opportunity to speak to you as representing MIT in this iconic voice series. The topic of my lecture is neuroscience of memory. Now, most people think about the memory when they remember the childhood, favorite childhood experience, such as riding a bicycle for the first time, or when one is feverishly looking for the misplaced the bunch of keys. But memory is much more than this. In fact, your memory is nothing but your life. The person who is unfortunate enough to suffer from serious dementia, like Alzheimer's disease, they may not even remember who he or she is. Okay? So some of the Alzheimer patients sometimes ask people around him or her, and they say, could you please remind me who I am? Okay? So the life, the memory, your memory is nothing but yourself. So this is the reality of memory. Not surprisingly, many well-known people made interesting remarks about the memory. For instance, Aldous Huxley, shown on the left side, said, every man's memory is a private literature. Bob Dylan sang, take care of all your memories, for you cannot relive them. Writer Haruki Murakami wrote that the memories are what warm you up from the inside, but they are also what tear you apart. So in understanding how memory works, let's go back for a moment to bigger issues of mind and the body, or you can say soul and the body. Because after all, memory is a typical example of a phenomena of mind. On this issue, the 17th century French philosopher René Descartes, whose portrait is shown here, claimed that people's bodies are like machines, capable of being studied by the method of the physical sciences, like physics and chemistry. But the soul, the most valuable and unique human attributes, lay beyond the reach of scientific method and could be approached only by rational reflection. This, however, this molecular, this philosophy, which is called the dualism philosophy, has been refuted by the 20th century revolution of molecular biology. And today, most scientists, particularly neuroscientists or brain researchers, believe in the opposite philosophical view that is called monism. This is typically expressed in the book which is published some years ago by Francis Crick, and you probably know who Francis Crick is, in the book called The Astonishing Hypothesis. In the first page of this book, Crick eloquently describes what is a human brain. Your joys and your sorrows, your memories and your ambitions, your sense of personal identity, and what you think is the free will are in fact no more than the behavior of a vast assembly of nerve cells and their associated molecules. The human brain is made up with 1 billion 10 to the 12 neurons, which are supported by several times more cells called glia cells. These neurons form a gigantic network through a structure which is called synapses, which is shown on the left lower side, through where the electric signal which pass through the nerve fibers is converted to what is called chemical signals. And this is again converted back to electric signal and run the fibers of the next neurons. Function of a neuron depends on a diverse set of macromolecules, such as the DNA, RNA, and the proteins. as well as small molecular weight chemicals. Furthermore, neurons are organized into a multiple layer of structural and functional complexity. From starting with a single neuron to a pair of neurons and to a functional brain system, such as structure called the hippocampus, and then to multiple function systems which communicate to each other, and finally to the whole brain. And when the organizational complexity goes up one notch, then new function is created, which is more than the sum of the function of the lower the complexity. So going back to the memory question, the fundamental question is how a memory is stored in the brain, gigantic network, and how these neurons are different from other neurons which are not storing specific memory. The idea memory is stored in the brain as the persistent physical and the chemical changes goes back to the time, or at least to the time of ancient Greece, the period of Plato's. But modern formulation of this hypothesis had to wait until the turn of the last century, 20th century, when Richard Zeeman, shown on the right side here, proposed, in fact, he coined the word engram for these presumed persistent physical chemical changes. But Zeeman's memory engram theory was entirely ignored by his contemporary and also subsequent brain researchers until early 1980s, when Harvard psychologist Daniel Schechter revived this theory by publishing this book, Forgotten Ideas, Neglected Pioneers. According to Zeeman's theory of engram theory of memory, for a population of neurons to qualify as a memory engram cells, at least 3 conditions have to be met. And this is listed in this slide. These cells have to be activated specifically by having particular experience. Now, second, in these cells, there has to be establishment of what is called engrams, which is enduring physical or chemical changes. And the third important condition would be that recall of that particular memory is accomplished only by reactivation of these cells which were activated originally by experience. And this can be done only through the action of the specific stimuli which was, for which memory was formed. And these stimuli are called recall cues. However, these studies have been very difficult to prove how the memory engram cells exist in the particular population of neurons was very difficult to do. The validity of this theory has been really formidable due to the insufficient availability of experimental tools and guiding conceptual framework to design an appropriate experiment. Nevertheless, it became gradually known that the area of the brain, which is called the hippocampus, which is shown in this image as a pair of horn-like structures on the left and the right hemisphere, shown in orange color here, plays a crucial role in the most common form of memory, like a memory of what happened to you yesterday, last month, or a few years ago. Okay? This was demonstrated in the middle of the 20th century by analysis of memory impairment of a single man whose name is Henry Moreson, which is widely known as HM. This handsome man, at the age of 9, suffered from brain injury in a bicycle accident, which led to a severe epilepsy. At age 27, neuroscientist Scoville operated on HM and removing area of the brain called the temporal, medial temporal lobes from both sides of the brain. Okay. That included the hippocampus that I mentioned. At that time, it was not known that the hippocampus played a crucial role for most modern type of memory. So, and it was the usual practice to remove that area of the brain by surgery in order to reduce the epileptic seizures. So this was done. And fortunately, good news was that the epileptic seizure was substantially reduced. However, soon people around HM discovered that he is suffering devastating learning and memory impairments. So he could not recognize people around him anymore, the places that he lived, or himself in a photograph, in a photograph or in a mirror. He could not remember what he ate for breakfast. Not only that, he could not even remember the fact that he ate breakfast already. So it was established that the hippocampus was crucial for memorizing what one encounters in a daily life. However, where exactly in the hippocampus the memory cells are located, and what physical and chemical changes have taken place specifically in those neurons as a result of the formation of a specific memory has been unknown until recently. So some neuroscientists felt that what could be the experiment that will really prove certain populations of neurons, like those in the hippocampus, really carry the memory information. So for this purpose, Martin and Morris suggested that something like what is written here has to be demonstrated. That is, the final test of any hypothesis concerning memory engram must be a mimicry experiment. Scientists should mimic what is happening during memory formation. So this experiment would require the memory is generated and expressed artificially without usual requirement of sensory experience. However, as I said, this has been very difficult because lack of sufficient technical tools and also the effective guiding principle to design experiment. In 2010s, early 2010, 3 people in my lab at MIT, Shuru, Steve Ramirez, and Arvind Govindarayan, decided to have a crack at this formidable task, being encouraged by the prospect of combining the cutting-edge molecular genetic technologies with a newly emerging technology called optogenetics. As a memory task for laboratory mouse, which we used what is called contextual fear conditioning, which is the rodent version of famous Pavlov's classical conditioning. So I'm trying to explain to you this behavioral paradigm. In this memory task, mouse is placed in a cage B, see on the left side, for several minutes. The mouse will explore the cage and form the memory of the specific feature of that cage. The mouse then receives a mild foot shock, as shown in here, that the electric shock, which is applied to foot pads, which is equivalent to encountering a predator in nature. It's a terrible experience. And the mouse will jump and scared and immediately form a cage B is a dangerous place memory. And this is a very acute memory. Once it's formed, it lasts a long time. The mouse recalls this memory on the next day if you put that mouse back to the same cage. And immediately after the mouse enters the cage, they show what is called freezing, the posture, which will, in nature, reduce the chance to be detected by predator. Okay? So this freezing can be used as an indication of memory. If you put the mouse to a very different cage on the day 2, I call it cage A, then of course cage A is very different from cage B, and therefore it does not remind the mouse the yesterday's terrible experience that happened in cage B, and therefore mouse will be happily run around. So here is the important procedure with which these 3 people discovered the memory engram cells for this cage B is the scary memory, scary place memory. We identified engram holding cell in the mouse hippocampus for cage-specific fear memory, and we could even engineer these cells artificially with light so that mouse recall the memory without the benefit of the specific recall cues. How did we do this? 3 important steps were taken. In the first step, as shown on the left side of the slide, we started with a transgenic mouse line called C-Force TTA. I will explain this in a minute. And surgically injected, as shown on the top left side, with a virus vector carrying light-sensitive protein called channel rhodopsin into a particular area of the hippocampus called dentate gyrus, which is shown on the left bottom side. We also implanted optic fibers just above the dentate gyrus of the hippocampus. These mice were kept in a particular context called the context A with an antibiotics called doxycycline in the diet. the C-force gene would be activated. It's known to be activated by running stimuli, in this case, electric shock to the foot pads. Now, this leads to activation of another gene called the TTA, tetracycline transactivator, which in turn activates channel rhodopsin gene expression. Okay. However, if doxycycline antibiotics that I mentioned is in the diet, this will suck up the TTA transcription factor, and therefore channel rhodopsin will not be formed in those cells. So only those cells which were activated by shock will be labeled with the channel rhodopsin, and those cells in the same area of the brain in the dentate gyrus which were not activated by shock will not be labeled with the channel rhodopsin. So, but now, okay, here we are. As the second step of this experiment, the mice were transferred to a contextual, the new cage called the context B. But in this case, the doxycycline was removed from the diet. And these mice received foot shock, electric shock to the foot pad. In other words, memory-inducing stimuli was delivered. Okay. This permitted the activation of a fraction of dentate gyrus neurons that were activated by the foot shock, and they are labeling with the channel rhodopsins by the mechanism I described a few slides back. And this shows that in the red-colored areas of the dentate gyrus are the one which shows the cell neurons that showed specific memory engram cells as a result of the formation of this cage-specific fear memory. The final step was the delivery of a blue light to activate artificially the labeled cell. And this was done in unconditioned, as shown on the right side, the bottom side, in a cage A, which is different from the cage in which the foot shocks were delivered. So you can see here that the consequence was a robust freezing of the mouse upon blue light delivery. In other words, record of the KGB specific fear memory without benefit of the natural recall cue derived from the condition KGB. The result fulfilled exactly the requirements which were imposed by the Martin and Morris that I described earlier. So here's a video, short clip, showing a mouse freezing when blue light laser is switched onto its genetically engineered mouse. Now, video comes in the two parts, first part and the second part. First part is what is called the control mouse. Mouse which did not go through this genetic engineering, and therefore, even if light is delivered, they should not freeze. And you will see this here. So initially, the light is not given, so animal would happily move around like you can see here. And now light is on. You see the light is flickering in the head. But the animal completely ignore it and move around without any problem. Here is the experimental animal that went through genetic engineering. Without light, animal will run around because now here come the lights. Now animal is completely frozen. It doesn't move around anymore. Now, as to the enduring physical or chemical changes which Demon engram theory demands. Remember, I told you that there are three conditions that have to be met. One of them is that there should be enduring, long-lasting physical chemical changes in those cells which are activated by during the process of formation of memory. Now, have we shown that? Yes, the answer is that Thomas Ryan and Michele Mignatelli and also Gerard Giroy found that when the mouse receives contextual fair conditioning, in other words, the electric shock in a particular context, an engram cell ensemble, engram cell population is quickly formed almost simultaneously, not only in the dentate gyrus, which is shown here, the DG on the bottom, but also the downstream hippocampal subfield called the CA3. And they also showed that the connectivity between the CA3 engram cells ensemble with upstream dentate gyrus engram cell is augmented as a result of the delivery of foot shock. Not only that, Michele showed this connectivity increase, augmentation of connectivity between engram cells populations extend further downstream of this pathway, neuronal pathway, including the CA1 area, and further downstream outside of hippocampus in the area called amygdala. Okay, so it looks like that the specific memory is stored in an enduring and augmented specific connectivity of engram cell ensemble that is distributed not only in one place of the hippocampus, but distributed in a multiple brain structures that include the hippocampus and the downstream amygdala. Now, this conclusion actually is consistent to Donald Hebb's decade-old theoretical hypothesis, which can be summarized in one phrase or one sentence. Neurons that fire together, wire together. And the brain uses these fundamental properties of, or features of neurons to store specific memory in the brain network. Since this initial discovery of engram cells in our lab, many labs identified memory engram cell in the various parts of the brain for different types of memory. It is not an exaggeration to say that these discoveries have created new area in the memory research. For instance, we can now not only make mouse to recall a specific memory at our will, as I described, but we can also artificially create a memory in the brain of a mouse for episode, for experience, which that mouse never experienced. Okay? So with no experience, memory is formed if you properly treat mouse. So this is the form of what is called false memory, not real memory, but a false memory. In other words, we can create false memory in the mouse brain. Many of you might think that memory is quite reliable, generally speaking, although we know that sometimes we make mistakes in remembering certain things that happened or not happened. In fact, it turned out false memory, human can make incredible false memory under certain conditions. There are many well-known cases like that. False memory could become a serious legal issues in a criminal court. When DNA tests were introduced, they revealed that an alarming number of prior verdicts that was made heavily relying on the memory of witnesses or victims turned out to be not correct. Okay. However, study on the cause of false memory have been hampered, primarily because animal model for false memory was not available, has not been available until recently. Now, we can now implant a false memory in the mouse brain. So this slide tells you, this is a cartoon, but it tells you the essence, the procedure of this false memory producing experiment. On the left side, mouse is placed in a blue cage, particular cage, so that it will form a memory of that blue cage, which will be labeled with the channel rhodopsin. Remember, no foot shock is delivered at this stage. On the next day, a mouse is transferred to a very different cage, red cage. And then blue laser light is delivered in such a way that the mouse can recall the blue cage memory, which was labeled with the channel rhodopsin yesterday. Okay. But at the same time, the mild foot shock is delivered. Okay. And if you do that on the next day, the third day, in the blue cage, mouse, as soon as the mouse enters the cage, mouse gets scared. Now remember, this mouse has not been has not received the foot shocks in a blue cage, and yet the mouse gets frozen. Okay, so this finding can be explained as follows. The mouse was recalling the memory of the blue cage on the day second, which not have been scary by itself. However, this recall occurred at the same time as the foot shock was delivered in another cage, the red cage. So the mouse falsely thought that it was being shocked. It was shocked in the blue cage. So these data on mice suggest that under certain conditions, humans associate an internally recalled memory of a past experience, benign experience, with a currently occurring major extreme external event, and therefore form a memory of an episode that actually never occurred to that person. Another example of the use of optogenetic memory engram cell identification and manipulation technology is the amelioration of depression. Depression is a terrible disease which afflicts 350 million people worldwide, and is often caused by chronic stress that lead to a series of what is called the negative valence memories, like fear memory or anxiety memory. Indeed, episodic memory, memory of things that happen to you on a daily basis, which is episodic memory, often has an emotional component. That component could be a positive one, pleasurable one, or can be a negative one, for instance, a fearful one. My former graduate student, Josh Kim, found there are two types of brain cells in another part of the brain called the amygdala, which I mentioned earlier. One type, stained green here, hold negative valence memory, like a fear memory. And then the other one, the other type, which is stained red here, actually contains the more pleasurable memory. Furthermore, Josh and Michele and also Roger, they discovered that these two types of neurons in the amygdala, which hold the positive, negative, the positive valence memory, they compete each other. Okay? If the positive valence memory is strong, the negative memory is reduced, and vice versa. So recently, we have made an exciting discovery, namely attenuating depression behavior in mice by using optogenetics. By optogenetically stimulating engram cells holding pleasurable memory. And it is known that one of the behavioral features of a depressed patient, now I'm talking about a human patient, is the inability to overcome or cope with challenging situations. Likewise, in case of a mouse, if one hangs a mouse by a tail, mouse doesn't like it, and therefore normal mouse would try to right himself or herself. And they struggle and try this, and they usually succeed within a minute and a half or so. But depressed mouse fails to do that. They will give up in less than 30 seconds. Another feature of a depressed patient is called anhedonia, the inability to seek pleasure or enjoy pleasurable experience. Anhedonia in depressed mouse can be measured, as shown on the right side, by making a mouse a little bit thirsty and providing 2 bottles of water. One is contains sweetened water, the other one regular water. Normal depressed mouse, thirsty depressed mouse will 90% of the time prefer to drink water from the bottle that contains the sweet water. But the depressed and thirsty mouse do not distinguish the water from 2 different sources. They cannot enjoy, they don't seek the sweet water and enjoy it. Here's a cartoon depicting the depression curing experiment. A normal male mouse, we use a male mouse for this experiment. You will see it immediately why. Normal male mouse was allowed to form a pleasurable memory by being allowed to play with a couple of female mice. okay? And engram cells for that pleasurable memory formed in the brain of a male mouse was labeled with the channel rhodopsin by the method I described already. Male mouse was then subjected to chronic stress by being repeatedly subjected to confinement treatment. Okay. And after a few days, when these mouses, the depression behavior was tested by the tail hanging test or the sucrose water test, it was found that these mouse will, if you give the light, blue light, while during a test, they behave like a normal mouse, okay, in both of these tests. In other words, depression seemed to be ameliorated by activating the mouse's the pleasurable memory engram cell by artificial optogenetic method. Here is a cartoon that recapitulates our depression attenuating experiment. Male mouse formed a pleasurable memory by interacting with, in this cartoon, one female mouse. And, and these cells were labeled with the channel rhodopsin. The male mouse then fell into depression by forming a negative valence memory caused by confinement-induced chronic stress. However, the depression behavior was significantly attenuated when the channel rhodopsin labeled pleasure memory engram cells were activated by blue laser light. Another important application, interesting application of the engram technology combined with optogenetics is a study on the mechanism of memory impairment associated with early stage of Alzheimer's disease. As is well known, in advanced stage of this disease, beta amyloid plaques appears in the hippocampus and elsewhere, mostly in the cortex, and that causes severe impairment in memory formation. But in an earlier stage of this disease, prior to the appearance of these plaques, beta amyloid plaques, in both human patient and mouse model of AD, it is known that already these people or patients or mice exhibit memory impairment, although plaques are still to appear. But my former graduate student, Dheeraj Roy, recently showed that this memory impairment is not in the formation of memory itself, but is in its inability to retrieve memory. Dheeraj further discovered that the reason of this recall failure in the early Alzheimer mouse model is that connection between the engram cell ensembles are too scarce. And Dheeraj could further overcome this memory recall impairment by increasing the strength of these connections, between engram cells, upstream and downstream, by shining blue laser light Apparently, he could even demonstrate, and amazingly, that such a treatment, artificial treatment, will even cure the inability to recall memory by natural recall cues. In other words, in this mouse model, early Alzheimer's, their memory impairment is more or less cured. So I would like to summarize what I've been telling you about our recent memory research. And there are 4 points to make. We can now identify brain cells that hold a specific memory, what is called the memory engram cell, by optogenetic technology. Number 2, a specific memory is stored in a specific connectivity pattern of upstream or downstream engram cell ensemble that is distributed in multiple brain structures, but not everywhere in the brain. In a particular different area of the brain, but the subpopulation of neurons form these memory holding cells. Third, we can now optogenetically manipulate the memory engram cell in the mouse brain so that even a memory of an episode that did not occur to the mouse can be created. We can ameliorate depression and the memory impairment associated with the early Alzheimer mouse by optogenetically engineering memory engram cells. Now, I would like to here talk about a little bit about the question of, is there any practical benefit? Are there any practical benefit from this mouse model studies on memory and memory impairment? And you don't have to read these very long sentences here. I will summarize for you. We have demonstrated what is called the proof of concept, or solution for the solution to depression and early Alzheimer's. We now know where in the brain the crucial problem resides in these diseases and what kind of problem are there. The connectivity of engram cells is abnormally reduced. Can optogenetics be a practically useful therapy for human patients? The answer is that it may be possible for disorders of what is called the peripheral nervous system, the nerve which runs through the body, okay, but not in the central nervous system within the brain. And this has been explored for instance, trying to cure blindness, particular type of blindness, which is called retinitis pigmentosa. And actually, this is actively being pursued in a biotech company with a great degree of promise. Okay. However, disease of the central nervous system, such as those like depression and Alzheimer's disease, is much more difficult, much more demanding. And I think we are going to need less or non-invasive technology that for this type of therapy. Okay. Now, I would like to end this issue with a positive perspective. So our hope is that non-invasive deep brain stimulation technology, like the one recently reported by my colleague at MIT, Ed Boyden, may develop into an effective therapy in the future. We hope time may come when entirely new type of therapy for these brain diseases that rely on physical technology rather than usual chemical technology based on the medicine or drugs. That time may come, which may take advantage of electric or magnetic technology, which will be applied from outside of the brain. So this is our hope into the future. So I would like to stop my talk here and just briefly acknowledge several people who contributed to this work. And memory engram discovery, it's already six years since this was first reported. And Shuru, Steve Ramirez, and Arvind Govindarayan. Memory, false memory inception experiment was done by Steve and Shu. And enduring memory engram cell discovery was done by Thomas Ryan, Michele Pignatelli, Diraj Roy. And the negative and the positive brain memory engram cell discovery was done by Roger Redondo and Josh Kim, which is another student. Depression engram engineering was done primarily by Steve Ramirez and Shuru. And finally, early AD engram engineering experiment was done primarily by Diraj Roy with the help from Michele Pignatelli and Thomas Ryan. Thank you very much for your attention.
Thank you, Professor Tonegawa. May I just invite you to take a seat at the middle of the stage? Well, it definitely brought back a lot of memories for me. I used to work on the hippocampus back in my postdoc days in the US. So once again, thank you for sharing with us your interesting studies. Hopefully, none of us go around shining blue lights on each other's brain. Yeah, you got to be genetically modified to do that, okay? So, well, I'm sure we're all definitely inspired, and there are tons of questions we're going to start asking Professor. But before we do that, we're going to invite on stage, we have our moderator from SUTD. He's the Associate Provost of Graduate Studies and the Co-Director of the SUTD MIT International Design Center, Professor Christian Wood. Let's welcome him.
So, may I transition a little bit here? Thank you. So, welcome audience, ladies and gentlemen, to SUTD. I hope you found that lecture to be riveting. With me, Professor, I will always have fond memories of your lecture, and I really appreciate that. It's a joy to have you, Professor, with us, and your wife, Mayumi, and experiencing Singapore with us. I know you actually even have a couple of your students, former students, in the audience here. So it's an absolute joy. So, as was stated, we are going to field questions during this session. You have a question right off the bat. And then what I'll do, if you don't mind, so I'll field your question, and then I have a question since I get to take the two. So you state it loudly, and then what I'll do is repeat your question for the professor.
Okay, thanks. I'm Bian Hongyu from NUS. Okay, thanks for your presentation. It's wonderful. I have two small questions. Firstly, why can external light activate the memory in some areas? Secondly, if the external can do it, can the part of synapse or neurons produce luminescence themselves?
So let me take them both. I'll divide them up. So the first question is, why the external light, blue light, can active memory in some areas? Yeah, so why can the blue light actually activate the memories, for example, during your experiments, Professor?
I'm not sure.
It is on.
Can you hear me? Okay.
Yeah.
Okay, I did not explain that part because time is limited. But this is the essence of what is called optogenetics.
Yeah.
So this protein called channel rhodopsin is a protein which is anchored on the membrane of the cell, okay? And if light is delivered to light of specific wavelengths, blue light is delivered, this channel opens and ions go in and out, okay? That is how the neuron is activated or inactivated.
Okay.
So that's the nature of channel rhodopsin.
Okay.
And the second part of your question?
Second, if the blue light can do it, can the internal parts of the neuron or synapse can produce blue light themselves?
Okay, so inverting the question, Professor. So if blue light can actually do the activation, can in the synaptic network, can it actually produce blue light? So I think you're trying to reverse it. Am I getting that right?
Can it produce the blue light? No. In the nature, this is not what is happening. Okay?
Happening.
Yeah.
Did I get your question wrong?
Yeah?
Okay, do it again, please.
Okay, thank you.
Thank you. Thank you very much.
Okay.
So I have a question for you, Professor.
Yeah.
All right, so there was a science article from some colleagues at Harvard.
Okay.
And in the title of the article is A Wandering Mind.
Wandering mind. Okay.
Is an unhappy mind.
I see.
So it's a little bit more global question.
Yeah.
Would you agree with that statement from the article?
Well, bottom line is, I agree. Okay? I agree. Wondering, if you are only wondering, and they doesn't know what to do this way or that way, I don't think that is very happy situation. It's a frustrating situation, okay? So, you know, we still a lot to learn about how human brain works, but nevertheless, first of all, from experience, my own personal experience, I can tell that if you are thinking about what to do, like in our research, and if I come to, there is a period where I don't know exactly which way to go, and it's very frustrating. But now there's something clicks in my mind, and I'm going to go this way and try how it will work. Okay? So in other words, my goal is set, and my way to go, try to reach the goal is tentatively worked out for me. And that is when, and I start working on this, doing experiment, and that is when I'm most happy, okay? So, you know, it's known that when people have an aim or goal, then people can be quite in a happy state. And it's well known that the chemical called dopamine is released very much when your aim is being pursued, okay? And with a positive sense of hope. Okay. So it seemed to me, both from personal experience and the second of all, a little bit of science about the dopamine function, I think this idea of a wandering brain is not a very happy brain, and the one in which you are working for the specific goal, specific aim, is happier brain.
Yeah, absolutely. In this science article, for the average human being, our mind wanders about 50% of the time.
I see.
So we hope it's like you said in a directed way.
But I don't mean to give a long answer, but you know, on the other hand, the scientists need a time to wonder. You understand that? Every time, all the time, the big portion of your time, you're just playing with this telephone, and you're not really wondering. Then I don't think a new idea will come for the new experiment.
I agree with you. There's good wandering and bad wandering, perhaps. Thank you. We'll have another question from the audience. Perhaps on my right, all right, your left, a question for the professor.
Thank you, Doctor. My name is Frank. I'm a sophomore in engineering product development. I found the research in creating the false memories in mice very interesting. So far, it seems that the new research is limited to creating like associations between different memories, such as between like fear and the blue room. So I was wondering that in the future, will it be possible to create an entirely new memory with no association to previous memories? Say, for instance, will it be possible to allow a student to imagine a hypercube, for instance? Yeah.
So the question is, in some of the experiments that you showed,
Yeah.
you created a memory based on association to previous memories.
Right, right.
And the question is, can you create an entire new memory without this association?
Yeah, that's a good question. This experiment can be designed, but has never been done. That's the situation right now. Okay, so I think it can be done, but the answer, as I said, it has never been tried yet.
Well, so this is an SUTD student asking the question. I think he should design the experiment
Yes.
and run it. Don't you agree? So, great, thank you.
Think about how to do it. Yeah. It would be fun to think about it.
Great, thank you for your question.
Thank you.
Perhaps in the center here, if you have a question, please.
Yeah, Declan McFadden, I'm now retired and challenged all the time to remember my kids' names at this stage, but apart from that, my question is on your trials to ameliorate depression and Alzheimer's. Was that reversible or irreversible? So once you took away the stimulant, the light stimulant, did those mice revert to being depressed?
Yeah, so what he's asking is that when you ran your experiments in terms of the depression or the early stages of Alzheimer's, when you provided stimuli and you saw improvement, was that reversible or irreversible? In other words, right when you removed the stimulus, did they revert back to the state of depression?
Well, in case of depression, we know that our lighting effect lasts about 5 days. Okay. Now, beyond that, we have not pursued. Okay. But it's not just the effect that occurs only during the lighting. Okay. If you give the light repeatedly, then the next 5 days, their behavior looks more normal. Okay.
Thank you for your question. We'll go to a question on my left over here. Please real loud.
All right. Good afternoon, Professor. Thank you for your lecture. My name is Ryan. I'm waiting to enter college, and I'm not trained in science, but you have demonstrated how the use of light can be used to elicit certain responses in the brain, and so I'm wondering whether it's possible for optogenetics to be used in media and advertising, where like certain colors and images can be used to provoke certain emotions or associations in the brain on a large scale. Thank you.
So the question is, and this is an entrepreneur. All right. Yeah, no, but I'm joking. So his question is, can now we start thinking about the possibility of using this for advertisement and other media to actually affect people and their responses to advertisement and media through the study?
You mean in terms of use of optogenetics?
Yes.
Is that what you're thinking?
Yes.
Yeah. As I try to explain to you, the feeling we have now among the people who are using the real optogenetics experts like Carl Deitroff, Ed Boyden, opinions is not identical. It's not the same. But the cautious view is the one that I described, that it's probably not for the disease of central nervous system. This technology, optogenetics itself, is probably too invasive, okay? And therefore, it will not become a very useful therapy. But if the nerve is not only in the brain, but you have a nerve in the whole brain. And eyes is a direct contact to the central nervous system, okay? But it's an end of that central nervous system. It's exposed in the eye. And there has been a serious effort to try to remedy the specific type of the blindness. And this seemed to be going quite well. Okay. So we'll see, I mean, how it goes. I think the people have not really given up. Some people are not giving up idea of even modifying optogenetics to the to improve the central nervous system impairment.
Interesting. So the periphery nervous system or peripheral nervous system
Yes.
is what you're talking about. Can you imagine too, sometime in the future, where like chronic stress, rather than treating it after the depression,
Yes.
being able
Beforehand,
to treat exactly because there's certain jobs.
Preventive medicine.
Exactly.
I don't know.
Perhaps an extension.
That is probably more difficult.
I hear you.
Yeah.
Great.
Yeah.
Thank you, Professor.
Yeah.
Thank you for your question.
Thank you, sir.
So let's go to my right here. Please, real loud.
Yeah. Hi, Professor. So I'm an alumni from SUTD as well. So it's a very fascinating talk. I actually have a question about so you've said that it is like a specific memory is formed by a specific conductivity of neurons. So what if I actually take away say, for example, a series or even a single neuron from that conductivity? What will happen to that memory? Will I still form a wholesome picture of that memory, or would that memory be altered by just taking away a single neuron?
So if a cell, we lose a cell, or we lose a series of cells that had formed a memory,
Yes.
is there ability to still have that memory within the brain?
Well, there may be some compensation by still alive cells, but essentially, if you kill the cell, or if cell dies, it's not possible to repair unless you do some kind of this stem cell approach. Okay.
So our memories then, are they clearly identifiable as a series of cells?
Yes.
And there is no replication of that memory in other places?
Well, there are actually. I mean, so I didn't talk about that issue. But if you look at the memory has a different degree of longevity, life. Okay. In fact, memory which you form today, and in the next few days, or maybe even a week or two, you hold it in the hippocampus, in the area hippocampus that I described. But this is not the final permanent site of the memory storage. Some of these memory can last a long time, 2 months, 3 months, 2 years, 3 years, sometimes a lifetime. And they are not in the hippocampus. They are transferred to an area called prefrontal cortex or neocortex. And those memory are not necessary, just because the hippocampus cell die, it doesn't mean those long remote memory, remotely formed memory goes away. Okay? So yes, memory itself, not exactly the same memory, but the memory very similar to the one which you formed in the hippocampus actually still stay in neocortical areas.
Thank you, Professor, and thank you for your question.
Thank you.
Over to my left here, please, real loud.
Hello. Hello, I'm Matthew, and I'm a first-year student from Nanyang Technological University, Singapore, doing a degree in biological science. So the topic you talk about, depression, is very interesting to me. I previously did something similar with optogenetics in my internship, but actually using fruit flies. And so it was regarding activation of dopaminergic neuron to induce positive valence behavior. So I think there's some research out there concerning depression with unbalanced neurotransmitter concentration in the brain. So I'm wondering if you can actually put channel rhodopsins into dopaminergic neurons, then actually use optogenetic to activate the dopaminergic neurons to restore the balance of the chemicals in the brain, or somehow remotivate the animal to seek pleasure again. Is that possible?
So I'm going to rephrase this. If I don't get it, you're going to correct me, okay? That was long. So as you put the blockers in, as part of your experiment, you put blockers in.
Yes.
And that's a chemical effect.
Yes.
Can you then actually have another intervention where you reduce that blocking to then regenerate or create the memory that was there, even though it was blocked originally? Did I do that, or did I do a bad job?
It's because there's some, how do I put it? Depression is due to like certain levels of dopamine like being low, right, in the brain.
Yes,
so I'm wondering if you can actually use optogenetics to promote dopamine secretion in the brain to actually remotivate the animal again.
Very good. So during depression, dopamine levels are depressed. Is what is being proposed here?
Yes.
Is there possible to use the approaches that you have here to actually increase dopamine levels?
So that, there's not only, I'm not saying that there's only one way that you can try to reduce the depression, okay? So yes, sure. There is also, I'm sure, other pathways you can use, like manipulating dopamine generating cells or connection between dopamine generating cells to the memory engram cell ensembles.
I see.
Yeah, there are multiple ways to try to address the problem.
And can those be produce long-term effects of perhaps removing depression?
Perhaps, yes, perhaps.
But no experimental results on that side yet that you're aware of?
Well, I don't know any specific the cases that were demonstrated by manipulating the dopaminergic system.
Very good.
Yeah.
Great. Thank you, Professor, and thank you for your question. We'll go to my right here, please.
Hi, good afternoon, Dr. Tonegawa. Thank you for the great talk. So I'm Fang Chen from UNS College, and we've been talking about optogenetics, but I have a question about memory engrams. So you've demonstrated that there's huge potential for memory engrams as like therapeutic targets for neurophysiological diseases. However, there doesn't seem to be a lot of labs that are focusing their research in this area. Do you have any explanation why is that so? Are people still skeptical about the engram and about what exactly is the content of the neurons that you managed to activate in this memory engram?
And I missed the last part of your question. One more time.
People are still skeptical about the existence of memory engrams and the content of the neurons that are activated in this engram.
I see. So just the extent, what is the extent of the engrams in their existence? And is there...
How long it lasts?
Or how prevalent they are? Is that a fair way to look at it?
Like, do people, how much faith do they have in these constructs as engrams that they have the potential to trigger memories that's beyond...
Okay. So through your work, has this work really resulted in people's understanding and it's being a definitive that engrams have this purpose, that they actually exist?
Yes.
Et cetera. Is it now relatively definitive in the field, or is there any skepticism?
Well, I think the fact that this concept of memory is held in a specific cell ensemble and their connections with other ensemble, I think this is quite well established now because it's been confirmed by many other labs. Okay, so the only question is whether some detail of what kind of changes on the molecular level takes place in these engram cell versus non-engram cells. And also the engram cell has a different state, multiple states, okay? So also the hippocampus versus prefrontal cortex, engram are formed in both places, but their properties are different. So the one in the prefrontal cortex takes care of very long-term memory. And the engram in the hippocampus takes care of relatively short-term memory. And they have a dual, apparently these two types of engram cells are cooperating each other to take care of the early and medium and the late stage of the memory. So there are many more things to be done with respect to really understanding exactly how, what the nature of these engram cells are at the molecular level, and also how their engram cell function is regulated in terms of the length of the memory.
Thank you, Professor. I hope did I address it? You want to follow up? Thank you very much. We'll go to my left here.
Hi, I'm Jeff. I'm the founder of an artificial intelligence company. I have two questions. The first question is about the patient HM. As I know, patient HM, after his hippocampus was removed, he still know how to play chess. He could play chess even after his hippocampus was removed. These skills actually was stored in his very long-term memory. I would like to know how did he record this long-term memory without hippocampus?
So the question is HM.
Yeah.
Right, HM, after he went through the surgical procedure, right. He could still play chess.
Yes.
And what would be,
yes.
What's the connection here versus things he couldn't?
Good question. So HM, analysis of HM, actually really revolutionized the memory research, okay? This is the first time, there are several things I can tell you. First, before HM, people could not even distinguish memory versus other intellectual ability, okay? So one could not conceptualize the differences between the memory, and if you have a good memory, you are very intelligent, and vice versa, okay? That's the way people thought. But now, with the HM, this type of memory, like episodic memory of that happens to him, it clearly he cannot show the memory, but his intelligence is very normal. Okay, his IQ test, if we do the IQ test, he was like nearly 100. Okay, so the intelligence and the memory are very different things. This is what was discovered. Okay. There's also different types of memory, and because the HM can do other things like require memory, like what is called motor memory. HM was not tested on this, but it's a similar kind of motor memory. And if you play tennis, if you learn how to play tennis, for instance, initially you don't do very well, okay? But you practice it, and you could become a very good tennis player. And that is a memory. Your body memory is the brain and your arms and the joints and the muscles memorize how to move your racket. Okay? So that's the type of memory which is very different from the memory of episodes. And HM can do motor memory perfectly well compared to this type of episodic memory. So this is the first time clearly there are different types of memory exist in the human brain. So there are many things, several things were really discovered because of the analysis of a single patient.
Thank you, Professor. Thank you for your question.
Thank you. My second question is, now there's a common sense that dream can improve the quality of memory. May I know how can dream improve or enhance our memory?
So make sure I get this right. So how can our dreams improve our memories?
Yes.
How can our dream state? Dreams.
Yes.
Improve our memories.
Yeah, this is good. This is a very hot subject. In fact, there are some scientists, like my colleague at MIT, Matt Wilson, is the front runner on this subject. And basically what sleep does, certain state of sleep, is called slow wave sleep. Apparently what's happening in the hippocampus is that specific kind of the oscillatory activity of a bunch of neurons is repeated in such a way that the celled neurons that hold the memory during a awake period, right after you acquire it, is repeated. That program of the memory firing is repeated during slow wave sleep. And when things are repeated, then connection between neurons become more tighter, okay? And therefore, this process is called memory consolidation. So sleep seems to be involved in a memory consolidation, which makes memories stronger or last longer.
So it's a form of reinforcement in some sense. Great.
Yes, right.
Thank you,
That's
Professor.
right.
Again, thank you for your question.
Thank you. Thank you.
I'm going to go. Unfortunately, this will be our last question. Fortunate for you, because of the time here, and we really appreciate the professor's patience with us. So one more question.
Hi. Thank you for the lecture, Professor. My name is Sid, and I'm from Tangling Trust School. And my question is, are there any adverse effects to any other parts of the brain due to the light or any of the biochemicals?
Yeah. So in the process here of perhaps doing treatment, the light, use of light, or other mechanisms to actually treat the cells.
Yeah, right.
Would there be any other adverse effects that would be expected?
Oh yeah, I'm sure there are some adverse effects. You can't have any kind of treatment that doesn't have adverse effect. The question is it's a balance between the positive effect and the side effect. Okay. Right now, even for disease, the Parkinson is another serious brain problem, brain disease. But the patients who cannot respond to the available medicine, they will volunteer to do invasive stimulation, electric stimulation, very deep inside the brain. And it still get some benefit of this, although there are also some side effects. Like people become, I was told, people become more gamble loving. Okay?
I understand.
They don't understand the mechanism itself, but there are side effects. So the question is, all these therapies are, in the medicines also, I mean, there's always some kind of side effects. So it's a question of what is the benefit compared to the side effect.
And then attempting to limit those side effects
Yeah.
as we study it more. So
Right.
Thank you for your question. Well, ladies and gentlemen, we've come to the end of our event. I hope you enjoyed the lecture. I hope you enjoyed being able to interact with the professor. And could you please give him our warm appreciation?
I want to give Chris for this amazing moderation.
Well, thank you both gentlemen. On behalf of our SUTD family, may I invite Professor Wood to give a token of appreciation to Professor Tonegawa.
So, let me make sure. Professor, again, it's been an honor and a pleasure.
Sure.
As a small token of our appreciation, thank you for
Thank
being
you
with
very
us
much.
Thank you.
At the Iconic Voices. Thank you.
Thank you.